Let's be real, identifying gram-positive cocci in the lab can feel like you're trying to memorize a phone book. It's just overwhelming, right? But what if I told you it's not about memorization at all? It's actually about following a logical, step-by-step map. So let's dive into that diagnostic roadmap. In this explainer, we're going to walk through that exact map together. We're going to take this seemingly complex web of organisms and turn it into a series of simple, smart lab decisions. And the best part? Each decision connects directly to taking care of patients. So here's our plan. We'll kick things off at the starting point, the gram stain, then we'll hit the great catalase divide, explore the two main roads, the staph path and the strep path, and then, at the end, we'll put it all together to master the map. Okay, so every journey has a beginning, and in microbiology, that beginning is almost always right here, under the microscope, looking at a gram stain. This is where we get our first big clue. The way the cells are arranged, we call that their morphology. Basically tells you which path you're probably going to be heading down. If you see those classic grape-like clusters, you should immediately be thinking staph. See those really perfect little squares, groups of 4 called tetrads? That's a good hint for Micrococcus. But if you see pairs or maybe these long snake-like chains, that's your signpost for the Strep and Enterococcus families. So with that first clue in our back pocket, we arrive at what is easily the most important decision point on our entire map. This is the first major fork in the road, and it all comes down to a really simple test, the Catalase test. And really, it all boils down to one simple question. When you take a little bit of your colony and you drop some hydrogen peroxide on it, do you see bubbles or nothing? And this is what it looks like. If you see rapid, fizzy bubbling, that test is positive. That sends us down the road to Staph and Micrococcus. If you just get a flat, sad little drop with no bubbles, the test is negative. And that steers us down a completely different road for Strep and Enterococcus. So think about it. This one simple test just split our entire universe of possibilities in half. It's huge. All right, let's go down that first path. We saw bubbles, so we're on the Catalase-Positive trail. This is the home of the Staphylococci. So what's our next move? We ask another critical question. Is it Coagulase-Positive? This test just looks for an enzyme that can clot plasma. If you see clumping on a slide, or even better, a solid clot in a test tube, you have just identified the heavyweight champion of pathogens, Staphylococcus aureus. And clinically, this is a really big deal. We're talking about wound infections, pneumonia, and of course, this is the bug we test for resistance, looking for MRSA, or methicillin-resistant Staph aureus. But what if that coagulase test is negative? No clot. Well, we move on to the Novobiocin susceptibility test. This little disc is amazing for telling apart 2 other key staph species. If you see a big zone of inhibition, meaning the bacteria are sensitive to it, you're probably looking at Staphylococcus epidermidis, that classic skin organism that loves to form biofilms on catheters and implants. But if it grows right up to the disc, completely resistant, that points you to Staphylococcus saprophyticus, a notorious cause of UTIs in young women. Now, before we leave this road, a quick heads up. You gotta watch out for Staphylococcus lugdunensis. It's a mimic. It can cause infections just as bad as S. aureus and can sometimes look coagulase positive on the slide test, so you gotta be careful. And then there's the other guy, Micrococcus. It's catalase positive, sure, but it usually has these bright yellow colonies, forms those perfect tetrads we talked about, and it's susceptible to bacitracin, which helps us sort it out. Okay, let's hit the rewind button. We're going all the way back to that catalase test. But this time, imagine we saw no bubbles. We are now officially exploring the Catalase-Negative branch of our map, home to the Streptococcus and Enterococci. On this side of the map, our new guide is something different. It's the pattern of hemolysis we see on a blood agar plate. We're looking for one of 3 things. Beta-Hemolysis is when the bacteria completely destroy the red blood cells, leaving a totally clear zone. Alpha-Hemolysis is partial destruction, which kind of looks like a greenish bruise around the colony. And Gamma-Hemolysis, well, that's just no hemolysis at all. This pattern is what's going to organize the rest of our trip. So let's start with those clear zone creators, the Beta-Hemolytics. Here, we can easily separate 2 major players. Group A strep, that's Streptococcus pyogenes, the cause of strep throat, is sensitive to a bacitracin disc and it's PYR positive. On the flip side, Group B strep, or Streptococcus agalactiae, is resistant to bacitracin, but is positive on a different test called the CAMP test. This is the one we worry about causing infections in newborns, which is why pregnant women get screened for it. Next up, the green zone, or alpha-hemolytic organisms. The key test here is another little disc, optochin. Streptococcus pneumonia, a huge cause of pneumonia, is very susceptible to optochin. But the Viridans group Streptococci, they're normal in our mouse, but can cause serious heart infections called endocarditis, are resistant. So again, one simple disc tells these two critical groups apart. It's that easy. And finally, we get to the Gamma-Hemolytic branch. No change in the agar at all. This is where we meet the really tough bugs that live in our gut, especially Enterococcus. So how do we nail this ID? Well, there's a three-part checklist. It's bile-esculin positive, which means it turns the agar black. It can grow in a really high concentration of salt, 6.5% NaCl, and it's PIR positive. Now, check this out. There's another organism here, the Streptococcus bovis group. Like Enterococcus, it also turns bile-esculin black. But, and this is a huge but, it cannot grow in that high salt broth, and it's PIR negative. Why does this matter so much? Because finding the S. bovis group in a patient's blood is a major clinical red flag. It is strongly associated with colon cancer. That's a finding from the lab that can literally save a patient's life. Wow. Okay. We've traveled down all the main roads and even a few side streets. So now it's time to zoom out, look at the whole picture, and really lock in the most important lessons from our journey. First, let's just quickly review those 3 critical red flag moments we found. You always have to be thinking about MRSA. You gotta remember that S. lugdunensis can be a really dangerous S. aureus mimic. And you absolutely cannot forget that powerful connection between the S. bovis group and colon cancer. And here it is, the whole thing, our complete journey, all in one view. You can see just how beautifully it flows. You start with what you see under the scope, the morphology. You make that one massive split with the Catalase test. And from there, each path has its own simple, powerful tests, like coagulase on one side and hemolysis on the other, that just guide you right to the final answer. So if you're going to remember just 3 things from all of this, make it these. Number 1, catalase is the great divide. It cuts the map in 2. Number 2, hemolysis is the main organizer for that entire catalase-negative side of the family. And number 3, a few simple key tests are what unlock these huge clinical stories that directly impact how we treat our patients. See, the moment you stop trying to just memorize a list of names and you start following the logic of the map, this whole process becomes so much easier and honestly, a lot more fun. If this explainer helped you out, make sure to subscribe for more CLS and MLT microbiology breakdowns from Lab Science Simplified. We'll see you on the next map.